Okay. We are going to continue with chapter 4. Announcements. So your assignment 1 is due next week. So I just answered some questions about the exam. Do you have any questions regarding the exam or assignment 1? You good? How many of you have read the exam review sheet yet? Not really? All right, you guys are pretty confident, right? Okay. Huh?
Is it on the campus page?
Yeah. It's been there for a week. What about the assignment? How many of you have started it? Okay, some of you.
I've looked at it.
Would you say it's time-consuming or hard or easy? What do you guys think? You don't know?
It's like every other project.
All right. How long do you think it would take for average students to do it? No clue? All right, just start early. Don't cramp it, because we can tell. I don't know if Emily can tell, but I can tell if I see your assignment, whether you cramped it or you actually put in the time, prepare for it. But just start early. All right. How many of you have study groups for this class? No one?
Not yet.
All right. So if you don't do well on the first exam, you can try it, or you don't have to wait until you've messed up the first exam. You can start a study group now, and then this definitely helps. Also, it keeps you accountable, set up study schedules. Just be careful when you have study groups. Don't get too close, I guess. Okay. I got an email about the psychology tutor. If you're interested, you can go to that link. It's $8 to $12 per hour. In my opinion, it's not that worth it, because you have Emily and I here. If you have questions, you can ask us. Unless you're like really struggling, I don't see the point going to a tutor. I don't know. It's up to you. Okay. Agenda. We are going to finish nervous system, and then we are going to talk about the peripheral nervous system today. So recap, the nervous system is the network of cells that connects one's body and mind. And it has 3 basic functions. First is to receive input from the environment, then it processes that input, and lastly, it generates output. Okay, so the building blocks of the nervous system. First, we have neurons. Those are the primary cells of the nervous system. They send and receive electrochemical signals. And there are 3 types. So there are sensory, motor, and interneurons. Next, we have glial cells. So people once thought they only serve minor functions, such as supporting neuron structures. They thought they were just there to hold other neurons together. But then they found out they actually serve more functions than that. So they direct growth of neurons, it provides nutrients, and restores damage to neurons. So for this chapter, we might not have as many discussions because it's biopsychology. I don't really know what we can discuss about other than just give you the information and trying to remember it. Sorry about that. Okay. Are we good on this slide?
Nope.
No? So a general tip is not to write down all the stuff that's on the slide. Instead, you should try to understand what I'm talking about, because you can get the notes afterwards. But if you know that you don't understand something, then you can ask questions. If you're copying it down, you're not really processing the information. You're just like writing it down. Maybe you think it's more helpful later on to look at the notes, but it's online, it's available. You don't have to write it out. So that will be a tip for you guys for the journal of the class, is to pay more attention to what I'm saying and try to understand the material. Okay. Okay, dendrites. Those are bushy branching extensions of a neuron that receive messages from other neurons and conduct them to the soma, which is the cell body. So those will be dendrites of the neuron. Yes?
So, like, does the neuron and the neuron have dendrites coming out of it that's, like, kind of like catching the information then?
Yes, so they receive information. What do you mean by catching?
Oh, receiving, yeah.
Okay. Yeah. They receive information. Yep. From dendrites. Right. Good. Okay. Next we have soma. That's the main body of the neuron. It's like the big part of the neuron. So they're responsible for life of the cell. So they are the metabolic center. The genetic material is stored in soma. Our information, which is neural impulses, is processed here in the soma. So that's the soma of the neuron. Then we have axon. That's this long thing here. That will be the tail of a neuron. This is the long tube-like structure that carries the neural message to be sent to other neurons. So they get information here from the dendrites, and then they process the information and then send it to this axon to the next neuron. Does that make sense? Yes?
So, does all neurons have their own, like, dendrite and axon, but they just, like, some, some neurons will, like, get one information and some won't, so that's why I need axons to be able to, like, transfer information?
Could you phrase your
So, like,
question a little bit?
if each neuron has a dendrite which is used to catch, like, messages and receive messages, then why do they need axons to be able to transfer messages if each one has their own dendrite?
So your neuron it's very small, right?
Mm-hmm.
It's not long. So if you want some information from your brain to your body, you can't just have one neuron and then transfer that information. So you have millions of neurons.
Okay.
This one transferred to the next one, and then the next one, and then from the brain to the body, or from the body to the brain. Does that make sense?
Yeah.
All right. Okay. So action potentials, they travel down the axon to the axon terminals, and we're going to talk about what axon terminals are. So myelin, those are the fatty coating on axons. They insulate, protect, and speed up the neural impulse. So more myelin equals more speed within a single neuron. Next, we have axon terminals. Those are the ends of the axon and the beginning of the junctions with other cells where messages are transferred via chemicals called neurotransmitters. So this is axon terminal. First, we have this knot thing. It is what we call a synaptic knot or terminal bottom. So those are rounded area on the end of axon terminals that contain synaptic vesicles. So synaptic, oh, this is so hard to pronounce. Synaptic vesicles are the little round thing here. So there are sac-like structures found inside the synaptic knob that contain the chemical neurotransmitters. So this gap here is the synapse. This is the tiny gap that neurotransmitters travel from the axon terminals to the next neuron's dendrites. So they're not touching each other. There's like a little gap between. Can you guys see the laser thing? A little bit?
If you,
'Cause
if you like hold it, yeah, if like you're moving it up.
Gotcha. I don't know if it's the light, because I thought this will be helpful for you guys to know what I'm talking about. But, all right. Any questions on the axon terminal structures? Good? Good. So electrical signal from axon triggers a chemical release from the axon terminals. And those chemicals are called neurotransmitters. And they're the chemicals that transmit the message on to the next neuron. For neurotransmitters, those are chemical messengers. They are released at the end of a nerve fiber, which is axon, by the arrival of a nerve impulse, and that would be action potential, which transfer the impulse to another cell. Different neurotransmitters have different effects. Some of them are excitatory and some of them are inhibitory. So when you're excited, you have certain type of neurons that excite you. When you feel blue, depressed, you have inhibitory neurotransmitters in the brain. Well, you have both at the same time, but sometimes you have more of the excitatory, sometimes you have more of the inhibitory. That's how your mood, your emotions are affected. Does that make sense? Yes?
This might be like a little off, but like, is this what like, you know, like if you take like different kinds of like drugs bad, is that what it does to your body?
So yes, so the different drugs have different functions. Some of them are mimicking neurotransmitters. Some of them tries to make your body to release those neurotransmitters. Does that make sense? Okay. Great question. All right. And then we have the receptor sites. Those are ports in the surface of the dendrites or muscles of or glands of the receiving cell. And different neurotransmitters, they have different receptors. And those receptors are shaped to fit a specific neurotransmitters. So the receptor site is like a lock and the neurotransmitter is like a key. If the shape doesn't match, you won't feel the effect of the neurotransmitters because the receptor sites did not receive that information. Okay. And that is why some drugs don't work because the patients do not have enough receptors. Let's say we are trying to treat depression. We might give them a drug that mimic, I think it's dopamine or norepinephrine to make people happier. But if they don't have enough receptor sites to receive that neurotransmitters, it doesn't matter how much drugs you take, it won't be effective. Right? So there are different reasons causing, let's say, depressive disorders or any kind of other disorders. That's why sometimes it takes a long time to find the right treatment for the client. All right. Questions on receptor sites? Good. All right. So this is the summary of the neuronal structure. First, we have the soma. It's the big part of the neuron. And then we have dendrites. That's receiving information. And we have this long tail, which is called axon, On the axon, we have myelin sheets. Next, we have the axon terminals. Those are here. On the terminals, we have vesicles. And then we have the synapse between this neuron to the next one. Okay, so that's what it looks like. Okay, so neurons. How do neurons communicate with each other? They communicate by transmitting electrical signals along their axons and sending chemical messengers into the synapse, and then to the next. So action potential. How many of you understand action potential? Did you guys read about it in the textbook? No? Okay. So this is kind of abstract. I'll give you the basic, what it does. So first, your dendrites get input, and then the information from dendrites, it goes to the cell body, which is soma. And then your action potential occurs. Those will be the electric signal. The action potential travels axon to synapse. From axon to synapse. And your neurotransmitters are released. I'm going to show you a video that explains much better than I do. And just pay attention to what he says and try to understand what a actual action potential is. You don't need to remember thickness. You need to put that down. So I was saying you need to pay attention to the video. You don't need to remember the numbers, like what's the voltage inside of the cell or outside the cell, but you need to understand how it works. Okay.
To like basic, like kind of like have an idea like the steps more than anything.
Yeah, so there'll be more details in each step here. This is like a summary, but he'll give you a much better demonstration. Okay. So it's quite long, so pay attention. I'm going to find a chair.
Before we delve into how neurons communicate, we gotta first understand a little bit of our old friend electricity. Basically, think of your body as a sack of batteries. I mean, you don't look like a sack of batteries. I'm just saying that your body as a whole is electrically neutral with equal amounts of positive and negative charges floating around, but certain areas are more positive.
You can't hear? Right. One sec.
Before we delve into how neurons communicate, we gotta first understand a little bit of our old friend electricity. Basically, think of your body as a sack of batteries. No, I mean, you don't look like a sack of batteries. I'm just saying that your body as a whole is electrically neutral with equal amounts of positive and negative charges floating around, but certain areas are more positively or negatively charged than others. And because opposite charges attract, we need barriers or membranes to keep positive and negative charges separate until we're ready to use the energy that their attraction creates. In other words, we keep them separated to build potential. A battery, just sitting on its own, has both a positive and a negative end, and the potential to release energy, but it doesn't do anything until it's hooked up to a flashlight or a phone or a kid's toy that lets those charges move toward each other, on the way converting electricity into light or sound or children's laughter. In much the same way, each neuron in your body is like its own little battery with its own separated charges. It just needs an event to trigger the action that brings those charges together. If you're thinking that this sounds more like engineering than anatomy, that might not be a bad thing. It might even help to think of your neurons in the same terms an electrician might use. Voltage, for example, is the measure of potential energy generated by separated charges. It's measured in volts, but in the case of your body, we use millivolts because it's a pretty small amount. In a cell, we refer to this difference in charge as the membrane potential. The bigger the difference between the positive and negative areas, the higher the voltage and the larger the potential. And just like there's voltage in your body, there's also current, the flow of electricity from one point to another. The amount of charge in a current is related both to its voltage and its resistance. Resistance is just whatever's getting in the the way of the current. Something with a high resistance is an insulator, like plastic, and something with a low resistance is a conductor, like metal. Now, when we talk about these concepts in terms of U, we're typically talking about how currents indicate the flow of positively or negatively charged ions across the resistance of your cell's membranes. And again, these membranes separate the charges, so they're what provide the potential to convert the electricity into something useful. Okay, now that we got Electricity 101 down, let's see how it works inside your nervous system. A resting neuron is like a battery just sitting in that sack that is you. When it's just sitting there, it's more negative on the inside of the cell relative to the extracellular space around it. The difference is known as the neuron's resting membrane potential, and it sits at around negative 70 millivolts. Where do those charges come from? Outside of a resting neuron, there's a bunch of positive sodium ions floating around, just lingering outside the membrane. Inside, the neuron holds potassium ions that are positive as well, but they're mingled with bigger negatively charged proteins. And since there are more sodium ions outside than there are potassium ions inside, the cell's interior has an overall negative charge. When a neuron has a negative membrane potential like this, it is said to be polarized. Now, these ions didn't just show up in this arrangement on their own. This is all orchestrated by one of the most important bits of machinery in your nervous system, the sodium-potassium pump. This little protein straddles the membrane of the neuron, and there are tons of them all along the axon. For every 2 potassium ions it pumps into the cell, it pumps out 3 sodium ions. This creates a difference in the concentration of sodium and potassium, and a difference in charges, making it more positive outside the neuron. This difference is an electrochemical gradient, and you probably know enough about biology by now to know that nature hates gradients. It wants to even out all of those inequalities in concentration and in charge to restore balance. But the only way to even out that gradient is for the ions to pass across the membrane. Thankfully, the sodium-potassium pump isn't the only way in or out of the cell. The membrane is also riddled with ion channels, large proteins that can provide safe passage across the membrane when their respective gates are open. And these gates open and close for different reasons depending on their structure and purpose. Most are voltage-gated channels, which open at certain membrane potentials and close at others. For example, sodium channels in your neurons like to open around negative 55 millivolts. But some others are ligand-gated channels. They only open up when a specific neurotransmitter, like serotonin or a hormone, latches onto it. Then we also have mechanically gated channels, which open in response to physically stretching the membrane. In any case, when the gates do open, ions quickly diffuse across that membrane down their electrochemical gradient, evening out the concentrations and running away from other positively charged ions. This movement of ions is the key to all electrical events in neurons, and thus is the force behind every single thing we think, do, and feel. Of course, not all of your body's electrical responses are the same, and neither are the flows of ions going in and out of your neurons. If only a few channels open and only a bit of sodium enters the cell, that causes just a little change in the membrane potential in a localized part of the cell. This is called a graded potential. But in order to send long-distance signals all the way along an axon, you need a bigger change, one big enough to trigger those voltage-gated channels. That is an action potential. And your best bet for making that happen is to depolarize that resting neuron. I mean, cause a big enough change in its membrane potential that it'll trigger the voltage-gated channels to open. It all starts with your neuron sitting there at resting state. All of the ion channels are closed and the inner voltage is resting at negative 70 millivolts. And then something happens. Some environmental stimulus occurs, say like a spider brushes up against a tiny hair on your knee, triggering those sodium channels to open, increasing the charge inside the membrane. Now, the stimulus and the resulting change have to be strong enough to cross a threshold for the true action potential to kick in, and that threshold is about negative 55 millivolts. Remember that number, because this is an all-or-nothing phenomenon. If the stimulus is too weak and the change doesn't hit that level, it's like a false alarm. The neuron just returns to its resting.
Okay, so remember the all or none phenomenon, but you don't need to remember the voltage. Does that make sense? Okay. I could try to explain everything to you, but I cannot speak that fast. So I figure it's more efficient for him to explain these things. What else? Okay, so you don't need to remember all those like different types, how they open the gates. What you need to know is why does it work, or how would I say it? Okay, think of it this way. If you can explain action potential to your friends in an easy way so that they understand, okay, so that's what happens in the neuron, that would be good enough. You don't need to go into like super details for the exams. Those will be more for the actual biopsychology classes, okay? But if you're not certain you need to ask me questions, you can do that too. All right.
State. But kind of like Doc Brown hitting 1.21 gigawatts in the DeLorean, once it hits that threshold, you're not going to travel in time, but you are going to see some serious action potential. At that threshold, the voltage-gated sodium channels open, and there are tons of these, so all of the positive sodium ions rush in, making the cell massively depolarized. So much so that it actually goes positive, up to about positive 40 millivolts. This is action potential in action. It's really just a temporary reversal of a membrane potential, a brief depolarization caused by changes in currents. And unlike graded potentials, which are small and localized, an action potential kicks off a biological chain reaction which sends that electrical signal down the axon. Because each of your neurons has lots of voltage-gated sodium channels, so when a few in one area open, that local current is strong enough to change the voltage around them, and that triggers their neighbors, which triggers the voltage around them, and so on down the line. As soon as all that's underway, the process of repolarization kicks in. This time the voltage-gated potassium ion channels open up, letting those potassium ions flow out in an attempt to rebalance the charges. If anything, it goes too far at first, and the membrane briefly goes through hyperpolarization. Its voltage drops to negative 75 or so millivolts before all of the gates close and the sodium-potassium pumps take over, things back to their resting level. Now, when part of an axon is in the middle of all this and its ion channels are open, it can't respond to any other stimulus, no matter how strong. This is called the refractory period, and it's there to help prevent signals from traveling in both directions down the axon at once. So that is the
Okay, remember that one. That would be important.
surprisingly simple app that your nervous system uses to let you experience the world. And because the voltages in this process are always pretty much the same, the initial threshold around negative 55 millivolts and the peak depolarization at 40 millivolts, your neurons only communicate in a single monotone buzz. It doesn't matter if it's a spider on your knee or an elephant, a paper cut or a stab wound, the strength of that action potential is always the same. What does change is the frequency of the buzz. A weak stimulus tends to trigger less frequent action potentials, and that includes if the stimulus is coming from you, like your brain telling your muscles to perform some task. If I need to do something delicate, like pick up an egg, the signal is low frequency. But a more intense signal, like trying to crush a can, increases the frequency of those action potentials to tell your muscles to contract harder, and the message turns into something that you can't ignore. Action potentials also vary by speed or conduction velocity. They're fastest in pathways that govern things like reflexes, for example, but they're slower in places like your glands, guts, and blood vessels. And the factor that affects a neuron's transmission speed the most is whether there's a myelin sheath on its axon. Axons coated in insulating myelin conduct impulses faster than non-myelinated ones, partly because instead of just triggering one channel at a time in a chain reaction, a current can effectively leap from one gap in the myelin to the next. These little gaps are the delightfully named nodes of Ranvier.
Alright, so another thing, if you want to control your muscles very intensely, the frequency of the action potential, is it high frequency or low frequency? His example was crushing the can.
High.
Okay, very good. So know that as well. All right.
Before we delve into
Okay, so let's do a practice question. When a neuron is at the resting stage, which side of the cell is negative? Okay, very good. So inside the cell, I can't explain this. You just have to memorize it. Okay. What does the myelin sheath do? Okay, very good. So speeds up the impulse transmission. Again, don't worry if you can't remember it now. If this is your first time seeing this, just go back and read through it. One way to check whether you understand this is to teach this to your friend or your family member. Once you are able to teach it, it means that you have a pretty good understanding of the concept. Okay. Any questions? All right. So what happens next is the nervous system. They have two main branches. First, we have the central nervous system. So this contains the brain and the spinal cord. Next, we have the peripheral nervous system. that's the nerves everywhere else. I think it's pretty easy to remember the difference. The brain, the spinal, the central, they're kind of in the middle, and everything else is the peripheral nervous system. Okay, so today we are going to focus on peripheral nervous system. I think we have enough time to finish all that. So peripheral nervous system, they transmit information to, and from the central nervous system. And this can be divided into two sections. One is the somatic nervous system, and the other one is the automatic nervous system. Okay. So we're down here. First, we have the somatic nervous system. That carries sensory information and controls movement of the skeletal muscles. And it has two pathways. The sensory pathway is from your senses to your central nervous system. So you get information from the environment, and then that carries information from your body part to your brain and your spinal cord. Then we have the motor pathways. That's from the central nervous system to your muscles. So after you receive the information from the environment, your central nervous system will need to give instructions. Let's say you accidentally touched a stove. Well, no, that's not a good example. Scratch that. Let's say I showed you a slide here. That's a stimulation, right? And you got the information. And then your central nervous system is telling you to move your hand to take notes. Right? That will be the motor pathway. Okay. Does that make sense? Okay. All right. Besides the somatic nervous system, we have the automatic. Oh no, it's autonomic. I keep pronouncing it wrong. So autonomic. Don't say automatic. Okay. So autonomic nervous system. This controls activities normally outside of conscious control. So this is how I remember it. Autonomic is something that we cannot control consciously, and somatic is movements and musculoskeletal movements, and those we can control. So this includes blood pressure, heart rate, your blood vessels, your pupil dilation, digestions, your organs, and your glands. So we don't really control those, right? Okay. So under the autonomic nervous system, we have sympathetic and parasympathetic. How many of you think this is confusing? Separating. Yes, Marcus?
You said how many of us think it's confusing?
Okay. Just Marcus? Yeah, a little bit.
I still do.
Oh, you still do?
Sometimes, yeah.
Yep. I think this is confusing too. Well, no, not anymore for me, because I taught this so many times. But I think it can be very confusing to someone who are exposed to this information the first or second time. It's like, oh, come on. I don't know how to make it easy for you to understand. So I remember when I was in undergrad, this is how I remember it. Is first we have the nervous system, and then we have the very important part, brain and the spinal cord. And then everything else is like about the body. So that's the peripheral. And you guys know what peripheral means, right?
Mm-hmm.
At that time I didn't. I had to look it up. By the way, when I was in undergrad, I had to look up all the terms and know what they mean. So peripheral nervous system is everything on the peripheral. And then we have somatic, which is controlling the muscle. It's like your peripheral nervous system controlling the muscle. And then the autonomic, it's the body part that you cannot control. After that, I'm going to explain what those are. Does that make it easier to understand or easier to remember? Okay, I hope it helps a little bit. Okay, so then we have the sympathetic nervous system. They mobilize body for actions, so they increase your cardiac output. That controls the fight or flight reaction, or sometimes we call it the 4 Fs. So the 4 Fs include fighting, fleeing, feeding, and mating. Okay. All right. So that's the sympathetic nervous system. Then we have the parasympathetic nervous system. So that reduces our bodily arousal. It conserves our energy. This slows our heart after we escape or in relaxation. And we call it the rest and digest system. Next, I am going to show you a chart, kind of summarize the difference between sympathetic and parasympathetic nervous system. Okay, so we understand it, right? The differences between. Okay. Okay, so here is a picture. You don't need to try to remember everything, but remember the main parts. So first, sympathetic. Your pupils dilate. It inhibits your salivation. So when you're nervous, when you see a threat, you're less likely to salivate. Okay? And then your bronchial dilates because it helps you breathe faster, get more oxygen, you can move faster. It inhibits your digestion because when you need to fight or flight, you don't want to digest, like spend energy on digesting. It stimulates glucose release by liver, so you need energy, which is glucose, to move your muscles or fight someone. Okay. After that, it stimulates epinephrine and norepinephrine. You don't need to know what they are. Just know they are excitatory neurotransmitters. So you remember excitatory and inhibitory?
Mm-hmm.
Okay. And then it relaxes your bladder, contracts rectum, because hopefully you don't poop yourself when you are very scared or nervous, right? When you're super nervous, you might lose that control. That's why we had the phrase, right? Shit my pants, right? Right? Okay. Okay, I don't want to discuss that. But if you're interested, you can take a look at that. It's pretty self-explanatory. Okay, so that's sympathetic, okay? And then parasympathetic, you constrict your pupils because you don't need a lot of attention. That's why you constrict it. And then it stimulates your salivation because you're relaxed, so you can eat food. And salivation helps with that. It kind of helps with digestion too. And then you decrease your heart rate. You have bronchial constriction. You don't need as much air. You're relaxed. And stimulates your digestion. Now you can spend your energy and time to focus on digestion. Stimulates your gallbladder. It contracts your bladder. It relaxes your rectum. So hopefully when you are using the restroom, you're not like super intense, right? You're pretty relaxed, and that's how I remember it. And then, okay. All right. Any questions on this? Does it help to remember the different nervous systems? Okay. All right, very good. All right, do you have any questions before we finish up? No? All right, let's do a practice question. I think this is the last one, I think. So while walking in the woods, Amy sees a bear. The sympathetic branch of her autonomic nervous system is activated. What will happen to her body? B. Okay, so she begins to breathe faster and her heartbeat quickens. Okay, very good. So B. So this will be the questions that you will see on the exam. Make sure you understand how they work and which nervous system is controlling what kind of activities in your body. All right, that's it.